Okay. So, maybe I should read the title since nobody else did. Hotspots and Mountains 2. We're going to pick up where we left off this morning for those of you who weren't. So, numerous previous studies have identified montane regions as areas of high species richness. Earlier today, we showed that in these areas of high richness, many small range species co-occur, and that the relationship of species richness to morphological and ecological diversity may depend on the turnover within cells. In other words, whether those species are ecologically similar or divergent. So this afternoon, we're going to focus on the role of topographic complexity in these patterns. Topographic complexity alone can generate multiple similar species, barriers to dispersal promoting speciation without differentiation, and increasing diversification rate and species richness, and then also decreasing geographic range sizes. Topographic complexity may also interact with weather or climate to increase environmental complexity. Different habitats on different mountain faces, potentially reinforcing physical boundaries, or accommodating ecologically differentiated species within small geographic ranges in areas of high species richness. And if those differentiated species are immigrants from other areas, high species richness and small geographic range size might not reflect high diversification rate. So when we consider all of these things together, we have sort of a general model that topographic complexity and climate predicts habitat heterogeneity or homogeneity, and that in turn predicts geographic range size and diversification rate, and that in turn predicts species richness. We have three model systems that we're looking at, three lineages of rodents, two primarily North American, Heteromyidae, that's kangaroo rats and pocket mice, and Marmotinae, which is ground squirrels and chipmunks. And then in Southeast Asia, we have Calisayurinae, which are basically all tree squirrels. Why do we expect differences among these regions? In Western North America, global cooling since the Miocene climatic optimum produced a general transition from forests to grasslands that was modulated by Pleistocene glacial cycles. So forests generally retracted at higher elevations, to higher elevations and higher latitudes, but moved downslope and reconnected across valleys during glacial advances, and then moved back upslope and fragmented during glacial retreats. Conversely, grasslands and deserts expanded at lower elevations and latitudes after the MCO and were fragmented by glacial advances and then reconnected during retreats. In Southeast Asia, we had quite a different phenomenon where most areas remained forested since the MCO, so that's over 30 million years of humid forest. But more open habitat expanded on the mainland as mountain uplift or lower sea levels isolated interior areas from monsoon rains. Those islands also experienced fragmentation when sea levels rose during glacial retreats. And so between lineages then, we also expect that in North America, marmotines and heteromyids occupy open habitats, except chipmunks, which favor woodlands. So we expect them to have different responses to the same climatic and environmental changes. And again, in Southeast Asia, the Calisayurines are occupying those very constant temporal tropical forests on both the mountains on the mainland and in the lowlands. So our data and methods. We have phylogenies from Menendez et al. For the squirrels and from Upham for the heteromyids. And in that case, we have 1,000 source trees, and we applied the maximum grade clitability function to reduce that, to get one best tree from that. The data that we're going to examine in this talk are the geographic ranges obtained from IUCN, projected to equal areas appropriate to those continents, divided into 25 square areas. We're counting species richness as the number of species occupying at least 25% of the area of the grid, each cell. And geographic range size is the mean range size for the species within a cell. We're looking at these geographic variables, topographic roughness, that's the largest difference in elevation of the focal cell from surrounding cells, habitat homogeneity, which is a measure of variation of the vegetation in a cell, accounting for leaf morphology, cover density, plant condition. Habitat count, so this is going to be heterogeneity, so it'll be the opposite relationship. The number of vegetation types with at least 50% prevalence in a 1 kilometer area within a grid cell. And then we have climatic variables. We have PC1 and 2 of the bioclim variables. These were calculated, the PCs were calculated separately for each of the areas. We also have one of those variables, the annual temperature range, the difference between the hottest month and the coldest month. And we also have growth season length, the number of days with temperatures above essentially 1 degree C, with no snow cover and water. And then our third variable is potential evapotranspiration, the maximum amount of water that could evaporate if there was water to evaporate. So that basically works down to the amount of energy, total energy inputs into an area. And these analyses were done in the EPM package. We performed multiple regressions using spatial autoregressive models to account for the spatial covariances in the error term. First, determining the combination of distance and neighborhood weighting schemes that provide the best model for the SAR error term. And in these analyses, the order of the terms does not matter. These are marginal sums of squares. Then we test for statistical dependence of species richness on each of these variables or on combinations of them, taking spatial autocorrelation into account. And also evaluate relationships of range size and diversification rate to habitat and climate. And then we take the outputs of those and put them into the structural equation models to evaluate claims of causal independence that are implicit in those combinations of linear models, and obtain from that also the standardized coefficients for the direct and indirect effects. So what do we got? So this is for heterogeneity. We have here the maps for topographic complexity, species richness, geographic range, and diversification rate. And what I basically want you to see is that the red areas are in different places. So if we're looking for a correlation between topographic complexity and species richness and geographic range, it doesn't look like there's a whole lot going on there for heterogeneity. We have a different pattern, but the same sort of incongruence between where the complexity is greatest, where the richness is greatest. We also see that the richness, as we could have seen in the other one, high richness over a range of topographic complexities. And we see highest geographic ranges consistently in these areas that are very low topographic complexity, but again, not uniformly the same complexity. And again, diversification rate is flat across the map. Calisayurni, again, we're seeing pretty much the same pattern. The species richness is greatest, not in the flatland areas, but clearly not in the Himalayas. And geographic ranges are largest in areas that have pretty low richness. And again, diversification rate is not doing anything. Well, but when we put those into the SAR models to account for the high degree of spatial autocorrelation amongst these areas that we were just looking at, we find that we do in fact have a lot of high correlations, very strong correlations, and there is just some variation in sort of magnitude and sign. And that's for species richness explained by topographic complexity, geographic range, and diversification rate. When we look at the effects of topographic complexity and climate on habitat homogeneity, geographic range, and species richness, we again see pretty much the same thing here in these linear regressions. I'm only showing you a few here. I won't numb you with all of these possible numbers that I could show. But again, what we're seeing is a lot of correlations, a lot of strong effects, and not a whole lot of information about how these might be interacting to actually causally explain the effects that we're seeing for richness. So here's our first preliminary structural equation. We're excluding habitat and climate and just asking, does topographic complexity explain geographic range, and that in turn diversification rate, and then species richness. And what you see is that across the board, topographic complexity has a weak to non-significant effect on geographic range size. Geographic range size has a huge effect on diversification rate in heteromyids only. These are not significant effects. And then diversification rate really only has a meaningful effect on marmotany. It has a smaller negative effect on callicyorines and a small positive effect in heteromyids. Now, these structural equation models then are not our final answer because, one, we have a lot of non-significant components here, and secondly, there are claims of independence implicit in this sequence that are not yet addressed. So when we do that, take those into account and adjust our equations, our models, accounting for that non-independence, what we get is that in heteromyids, topographic complexity has a direct but weak effect on species richness. Geographic range size has a stronger, also negative effect. We have a similar pattern really in marmotany, but there is also an effect of diversification rate. And when we get to callicyoriny, the only thing we see is a large negative effect of geographic range size on species richness. So that's the first part. Now, what we also wanted to know was what does climate and habitat do to modify those relationships? And again, I'm just going to skip to the end here and talk about the structural equations for habitat and climate. First, for heteromyids, we have that two of those variables do affect geographic range size, but with opposite effects. Evapotranspiration or heat influx has a negative effect. Temperature range, a large one. These are desert animals, so they're avoiding the wetter and more fluctuating, but have large range sizes in more northern areas. Geographic range size affects diversification rate and species richness, and growth season also has a direct negative effect on species richness in this group. In marmotines, it's a very different pattern, but what we see is that climate components have strong effects on habitat homogeneity, which has no effect on species richness. That bell is really deafening up here. And geographic range has a small independent, but it's not affected by climate. Callicyorines give us yet again another pattern where there's really just two strongly correlated climate variables that affect range size that affect species richness. So in summary, what is this? There's a negative correlation between species richness and the average geographic range size of the coexisting species in all three groups. There is not a consistent relationship between diversification rate and species richness. And the effect of climate differs among regions, lineages and regions, not legions. There are some caveats. The variables that we use are not necessarily the ideal ones. Topographic roughness is not the only measure of complexity. There are other climate variables that we could have considered, and these methods are still under development. What does that say about the relationship between complexity, topographic complexity, and species richness? There are other variables to consider here, especially I want to throw out the idea of thinking about continuity and connectivity. The continuity of the humid forest in Southeast Asia may account for the high species richness on the sun-to-shelf islands, higher than on the mainland. The connectivity of ridges and valleys in the Basin and Range and the Mexican Plateau may have facilitated range shifts during climate change and accumulation of species in areas of confluence. And because those lineages occupy different parts of the Basin and Range, their areas of highest richness are actually in different areas within that same montane complex. And so with that, I'll stop and thank all of our curators. And if there's a few seconds left, I'll take questions. Maybe not.
All right. right, without any further ado, this is a fairly long talk. I'm just going to be zooming in from the last talk down into Southeast Asia, but moving away from the highlands into the lowlands. I'm going to be talking about the origins and consequences of the bluewing pitta range expansion, but more broadly looking at diversification within a particular bird complex, the bluewing pitta species complex. Right, so I'm going to start with a very different species. This is the mangrove pitta. This is a species that occurs in mangrove swamps all the way from the Sundarbans in Bangladesh south to Sumatra, right? You can see the contemporary range is highly fragmented in part because of the loss of mangrove habitat throughout its range. And that, in turn, makes this species relatively threatened. I think the current status is it's considered near threatened across its global range. In Singapore, where most of the study was conducted, where I'm from, right, you can see mangrove loss has been pretty severe, and there is now an estimated 20 individuals left in the whole of Singapore. But we'll come to see that this number is a little bit probably going to be wrong in a few slides' time. Now, compare this with the bluewing pitta, and I promise I did not copy and paste the photo. Right, this is a sister species of the mangrove pitta. It looks really, really similar, and you can see from the Birds of the World map here that it occupies a much more inland range. Right, it occurs in tropical rainforests. It breeds in tropical monsoon rainforests up in Indochina, and it winters down in lowland Southeast Asia and Malaysia, Indonesia, Borneo, Singapore during the northern winter months. Now, in the last decade as well, we have started to see a slow range expansion of the species. From Indochina, in 2005, we first detected breeding in Taman Negara in Peninsular Malaysia, and in 2014, we had our first breeding record in Singapore, right, on this one island called Pulau Ubin, where the blue dots are. And if you compare, right, if you zoom into Pulau Ubin and you look at these blue ranges where the bluewing pittas are known to breed and the orange patches where the mangrove pittas are known to breed, they are now highly sympatric, right? They are literally side by side with each other. You can walk along one trail and hear bluewing pittas calling on one side of the road, and the other side of the road you hear mangrove pittas. And if you listen to the vocalizations, this is mangrove pitta. This is bluewing pitta. The bird watchers here will say this is night and day, right? But everyone else will go, what the hell is the difference, right? Pardon my French. And just looking at them morphologically, there's also not much differentiation going on as well. So when you have one species that is fairly threatened, right, coming into contact, secondary contact with its sister species with very little morphological or phenotypic differentiation, there is this really high risk of hybridization, and hybridization is more likely than not to harm the threatened species. And so this is a conservation concern. And so what we did was basically to try to sequence these birds to try to investigate, number one, whether there is evidence of hybridization between these two sister species, right? And also to look at the population genetic health, right? Genetic diversity of the threatened mangrove pitta, and also to try to figure out what the evolutionary origins are of this bluewing pitta range expansion. Is it due to migratory drop-off, right? Migratory birds coming down to the wintering zone and deciding, you know what, I kind of like it here, I'll stay here long-term and breed, right? And so to get at this question, we sampled birds from, we sampled mangrove pittas from Singapore. Unfortunately, we can't sample mangrove pittas elsewhere because of permit issues. We were also able to get both resident breeding bluewing pittas and migratory bluewing pittas from Singapore, right? Since we have both passing through Singapore, most of the migrants came from salvage window strikes, and the residents were misnetted in the field. And we also were able to get migratory bluewing pittas from a small island in Thailand, right, which will give us a nice way to contrast with birds that are much closer to the breeding grounds as well in Indochina, right? We use dd-RADseq to obtain SNPs, and these are the first results, right? So as far as hybridization is concerned, you can see from the PCA plot, can't see there, I'm going to move away, right? Can you hear me? Okay. The migrants cluster up here, both the Singapore and Thai migrants cluster up here, and the residents form this climb with the migratory bluewing pittas, right? And the mangrove pittas are all the way over here. So basically, we see from the PCA plot no evidence of hybridization, or it could mean that we just didn't catch any hybrids. But when we look at the admixture plot, we also see very minimal evidence of hybridization or any form of admixture between the mangrove, which is on the far left side, right side, and the bluewing pittas, which are on the left side, right? Running an ABBA test, we also see no evidence of introgression between the bluewing pittas and the mangrove pittas, which is really unusual considering how weakly phenotypically diverged they are, right? Now, if we plot, this is based on DD-RAD, so this is a really rough pseudo-GWAS type thing. We only sample about 0.8% of the genome. But when we plot these SNPs across the whole genome, we do not see any particular regions that are especially divergent, right? The divergence is about 0.4 FST, and it's fairly uniform across the whole genome. Of course, slightly higher divergence in the sex chromosome, which is expected, right? So that's part one, right? No evidence of hybridization. Now, part two, what about the mangrove pitta? What's going on with them, right? If you look at the heterozygosity, pi, which are measures of genetic diversity, as expected, the blue wing pitta has a much higher genetic diversity, both the resident and the migratory blue wing pittas, compared to the mangrove pitta, which is a much lower genetic diversity. However, the mangrove pitta, right, in the last bar plot there, has a much lower inbreeding coefficient, which suggests that the population is fairly outbred. Right? So there's a fair amount of movement of mangrove pittas between subpopulations. Right? When we go to the effective population sizes, we see that the inferred effective population size, this is inferred using BPP, right, which infers the effective population size, gene flow, and divergence times jointly, right? We obtain an effective population size for the mangrove pittas of 29,000, considerably more than the 20 that we initially estimated the population size to be, which suggests that this entire region in Southeast Asia, right, with mangroves, the Riau Islands in Sumatra and Singapore, are probably part of a single metapopulation, which means that there's probably gene flow between these areas and it's fairly well mixed, right? We know from data that I'm not going to show here from hand-ring index measurements that mangrove pittas are have a fairly high hand-ring index, which means they can probably disperse pretty well between areas as well. All right, so let's get into the origins, right? Where do the mangrove pittas and the blue wing pittas come from? Now, Mike Harvey in his 2020 Subausine tree paper suggested that mangrove pittas split from the blue wing pittas about 5 million years ago, 5.4 million years ago. However, our SNP dataset and the mtDNA dataset, part of which came from Mike Harvey's dataset as well, suggests that in fact the split between mangrove and blue wing pittas was much more recent, only about 500 to 600,000 years ago, which is well within the Pleistocene, right? Not in the Miocene as earlier thought, right? However, more interestingly, before I go to the next one, right? And why this might have happened, right, could be due to the way the extent of mangrove forests could have shifted during the Pleistocene. So here I'm plotting, right, a map of a predicted map of mangrove extents across Southeast Asia over the last 1 million years. And as you can see, right, in the Andaman Sea, where the core range of the mangrove pittas is today, right, the mangrove forests that have been fairly stable over time. So harking back to the previous talk about connectivity and stability, contiguity of forests across the Pleistocene, right? Whereas on the South China Sea side, you can see the mangrove forests are shifting over the last 1 million years, fluctuating like mad because of the sea level change and the interaction between sea level change and topography, right? And so coming to the blue wing pittas, it looks like the blue wing pittas are not just one population. It is in fact two deeply diverged populations. The resident population and the migratory population appear to have diverged about 500,000 years ago as well, fairly close to when the mangrove pittas split out from the blue wing pittas, right? About 511,000 years ago. And my conjecture, and we still need more data and more sampling for this, is that during the Pleistocene, right, there was a population of blue wing pittas down in the northern part of the Thai Malay Peninsula that got isolated when the Isthmus of Kra narrowed. And this narrowing of the Isthmus of Kra would have happened multiple times across the Pleistocene as sea levels rose and fell and rose and fell, right? And around 500,000 years ago, this population was, there was a break between these two populations, although we do infer a large amount of asymmetric gene flow from the migratory population into the resident population. So it is possible then, so therefore it suggests, right, that this colonization of lowland Southeast Asia by the resident blue wing pittas is not a case of migratory drop-off, it is just a range expansion of the southern population of the blue wing pittas, right? So conclusions. So as you can see from the graphics, no hybridization, right? Divergence and speciation in the blue wing pitta complex occurred around the mid-Pleistocene, much more recently than previously thought. It is possible, and this needs to be dug into much more deeply, adaptation to mangroves and maybe a semi-saline, subsaline environment could have resulted in strong reproductive barriers being evolved between the mangrove and the blue-wing pitta, right? And it is possible, again, this needs more work, the stability of mangroves in the Andaman Sea could have facilitated ecological divergence, right? And that the repeated narrowing of the Isthmus of Kra could have facilitated divergence of non-migratory blue-wing pitta in the southern part, in the northern part of the Thai Malay Peninsula, right? And as far as conservation is concerned, the mangrove pitta genetic diversity remains low. Whether this is due to contemporary habitat loss or because of historical mangrove dynamics, it's not clear at the moment. We need more data, right? And that the range expansion of the blue-wing pitta is not a case of migratory drop-off. It is potentially due to the expansion of dry monsoon rainforests down Thai Malay Peninsula, which facilitated the expansion of the species range. Right? Now, as far as the conservation implications are concerned, this is a much more applied site. The lack of hybridization is good news for the mangrove pitta, although this does not preclude interspecific competition where they are in contact. And mangrove pittas, since they seem to be able to disperse fairly well, recolonization of previously defaunated mangrove habitats could be a good indicator of recovery, right? And a lot of future work to be done. Are there islands of genomic divergence between the two species? We are working on getting whole genomes where we can dig into this question a lot more deeply. Are there genes associated with mangrove adaptation, salinity tolerance, maybe crab-dominated diets? We don't quite know yet. Also, are there genes associated with migration? And this is something that we're also working on very much so. And then what are the potential drivers of blue-wing pitta range expansion? We're going to be doing some niche modeling as well to look at this. And how has the historical biogeography of Sundaland affected speciation more broadly, especially in the continental part of Southeast Asia? We've seen some work come out of Scott Edwards' lab on the Ceyx Erytharchus complex, right? And so this is going to be a continuation of some of that as well. A lot of acknowledgements. This is fieldwork across 2 countries, a lot of collaborators, and I cannot name everyone, but thank you so much to my Thai collaborators, my Singaporean collaborators, and my colleagues here in New Mexico as well. Any questions? Okay. No questions? Yes?
Maybe I missed, maybe you explained, what was finally the consequence of range expansion?
We don't know. I mean, so we know the range has expanded, but as far as the consequences are concerned, we don't really have any, we don't see any hybridization, which is good news. But aside from negative consequences, we don't really know.
And what do you think, why the divergence time that you estimated was very different from the previous?
So UCEs tend to evolve fairly slowly, ultra-conserved elements, and so it might be pretty useful for dating backbone nodes, but I think more recent nodes will probably have some sort of bias that pulls it back towards the past. That's my conjecture, and people who do UCE work, feel free to correct me. Question?
Any thoughts on competition between the two now that there's some patchwork?
That's future work for future me. If not, thank you so much for listening to my very rushed talk.